Hey everyone, this lesson is on polygenic inheritance and epistatic interactions. When we did start off our discussion in genetics by talking about the work of Gregor Mendel and all the different traits that he did take a look at, each one of those different traits were controlled by a different gene, but each one of those only had two different possibilities, two different possible versions, or two different possible alleles. We later saw that in some cases we could have a gene that has more than two different possible versions or alleles. So one that we did take a look at was the human blood typing, the ABO blood typing system. And we saw that there are three different possible alleles for this one single gene. But now what we're going to talk about is traits that are controlled not just by a single gene and possibly more than two different alleles, but traits that are affected by more than a single gene. And that is where the name poly does come from, poly meaning many, and genic, we're talking about the genes. So keep in mind when we do talk about a gene, what we're talking about is a set of instructions that we do find at a specific location or locus on a specific chromosome. And if we do have a trait that is controlled by more than one gene, that means that they would have to occupy different locations. They could be on the same chromosome or they could be on different chromosomes. So we'll take a look at a few different examples of traits that are in fact controlled by more than one gene. So a few human examples. If we take a look at human eye color, this one is controlled by at least three different genes. We will take a closer look at human height. That one as well, probably controlled by three different genes. But just keep in mind that whenever we have any trait that is controlled by more than one, that is what is referred to as polygenic inheritance. So if we do take a look at the genes and the effects of these genes, they might interact with each other, and depending upon what the genotype is, what we can end up with is not just a few, but a wide variety of different and intermediate types in the phenotype that is produced, again, depending upon what the genotype is. So if there is a great deal of variation for a particular trait, then chances are it is likely to be polygenic. So I mentioned we would take a look at human height, and even though it's probably controlled by three different genes, I'm just going to talk about it being controlled by two different genes simply because we haven't talked about three trait crosses. We've only talked about two trait crosses dealing with two different genes at the same time with the two trait or the dihybrid crosses. So in the case of human height, it appears that each dominant allele, in fact, for all three of the different genes, each one of the dominant alleles, they have an effect that is additive. So more or less what that means is the more capital letters, the taller the individuals are going to be, and the fewer the capital letters, the shorter the individual is going to be. If you have a combination of capital and lowercase letters, well, you're going to be somewhere in the middle. So I mentioned there are, in fact, probably three different ones. On the example that I have on the next slide here, I'm just showing it for two different genes. Again, just so we are only taking a look at a dihybrid cross rather than a trihybrid or a three trait cross. So as I mentioned, it's the greater number of capital letters that do result in the taller individuals. So if we have two parents that are heterozygous for both of the genes, these two that I'm showing here, then they would indicate that both of these parents would be sort of intermediate in terms of their height. But keep in mind that each one of these parents, they can contribute gametes that can either be the two capital ones, the two lowercase ones, or one of each, a capital and a lowercase letter. And that would be the same for the second parent here as well. So I'm not going to set up the Punnett square for this one, but hopefully you can sort of piece together that it is possible for these two parents to have some individuals that some offspring that have all capital letters. So these ones here would end up being the tallest. There are some individuals that could possibly get all lowercase letters. These ones would end up being the shortest, but most of the individuals from these two parents, they won't have all capital letters, they won't have all lowercase letters. They will have a combination of capital and lowercase letters, and that is why from the two parents that would be heterozygous, we would get a pattern that would look something more like this. Now, I mentioned that in fact it's probably three different genes. So if we just use the letter C to represent a third gene that is also responsible for height, these individuals that are the tallest, they might have this combination: capital A's, capital B's, and capital C's. The ones that are truly at the other end would have all of the lowercase letters. And again, we can see here that now we have many, many different possibilities in terms of the height by using these three different genes. One other thing that I'll point out is that, of course, height is not just dependent on genetics, it is dependent upon environmental factors as well. Things like diet, of course, are going to have an influence on how tall or short that individual actually is. So we'll take a look at another example here. This one here is taking a look at the color in birds or parakeets. And again, there are two different genes that I'm going to talk about here. And we'll just call them the blue gene and the yellow gene, because what they do result in is having a set of instructions that either leads to the production of a blue pigment or a yellow pigment. So we'll start off by having the two parent genotypes here, where the first parent, this one is going to be blue in color, and it's going to be blue in color because they do have the instructions for the blue, the capital B, but they don't have the instructions for the yellow, the capital Y. In the case of this individual, they could be either homozygous for the capital B or heterozygous, and they would end up being the same color, blue in color, but I just made this one here homozygous dominant for the first, the B allele, and homozygous recessive for the second one, the yellow lowercase allele or the yellow gene. For the second parent, this one here, exactly the opposite. So this one doesn't have the instructions for the blue pigmentation, but they do have a capital Y. Once again, it doesn't matter if it's one or two. In this case, they are going to end up being yellow in color because they do have the capital Y, but they don't have the instructions for the blue pigmentation. So if we do take these two parents and take a look at the gamete genotypes that are going to be produced. So from this parent here, there's only one possible gamete genotype, and that's going to be a capital B and a lowercase y. And from the other parent, of course, the only possibility is going to be a lowercase b and a capital Y. So when those combine together in the offspring, we only need one square in the Punnett square, and all of the offspring in that next generation, the F1 generation, are going to be heterozygous for the B and for the Y gene. So in this case here, it just so happens that these individuals, they end up being green in color. What you don't want to confuse this with is what we have talked about before with incomplete dominance. So this is a blending, but it's not a blending as the result of incomplete dominance. When we talked about incomplete dominance, that is one single gene, and that one single gene having two or more possible alleles. Homozygous individuals will have one phenotype. Heterozygous individuals will have a different genotype, a different phenotype, which is typically an intermediate between the two of them. So this one kind of looks like that. We have the blue, we have the yellow. You mix them together, you do end up with the green, but it's not incomplete dominance because it's not one gene that we're talking about, it's two different genes. So it's not incomplete dominance, it is polygenic, two different genes contributing to the same phenotype. If we were to now take these two individuals from the F1 generation and cross them, then what we would end up with in our Punnett square for the F2 generation, we would have to use all 16 squares in the Punnett square. And what that would result in is a 1 to 3 to 3 to 1 phenotypic ratio in this case. I am going to draw this one out, so just bear with me for a couple of minutes as I go through and draw all of the gametes and all of the offspring that we're going to get from this particular cross. So again, from each one of the parents, we're going to get 4 different possible gamete genotypes. We can get the big, we can get the big little, we can get the little big, and finally we can get the little as well. So going through, I'll just fill all of these in for all 16 of the squares that we have here. We did see this before as well. What I said is there is kind of a reason for writing the gamete genotypes along the top and the side the way that I am showing it here. So starting with the big, and then the big little, the little big, and the little. It's just a little bit more easy to organize it at the end and pick out from the different genotypes what the phenotypes are going to be. And we'll see that in a minute. Okay, so now I'll use the green to circle all of these ones here, except for this one right in the middle. Okay, so all of those that I did circle in green, you'll notice that, well, there are 9 of them. And out of these 9, they all have at least one capital B. It doesn't matter what the second letter is. At least one capital Y. It doesn't matter what the second letter is. And all of them are going to be green in color. The one that I circled and these 2 that I will circle here as well, we have 3 of them in total. They all have at least one capital B, but they don't have a capital Y, and they're going to end up being blue in color. These 3 that I'll circle here in red, they're not going to be red, but we do have 3 of them. These ones have both lowercase b's and at least one capital Y. No instructions for the blue, instructions for yellow, so these ones are going to be yellow in color. So now the question is, the one remaining one that we have right at the lower right-hand side here, what color is this one going to be? We only have one of them, so notice this is a 9 to 3 to 3 to 1 ratio, that ratio that we've seen many times before with the 2-trait or the dihybrid crosses. This last one, in fact, is going to end up being white in color. So finally, what is the explanation for this? What is actually going on in terms of the instructions in the genes? What is produced from these instructions? And how does it lead to these different phenotypes that we're talking about here? Well, quite often, what the genes are going to be coding for is an enzyme. An enzyme, you might recall, is going to catalyze a specific biochemical reaction. And in the case of the parakeet color, what we have are 2 different biochemical or metabolic pathways that are involved. So the first one, we are taking a starting material or a substrate, and if the instructions are there to produce the blue pigmentation, then that pigment is going to be produced and that color is going to be visible. But in order for this enzyme that catalyzes this reaction to occur, you have to have at least one capital B. So if there's a capital B, this reaction takes place. If there's not a capital B, this reaction doesn't take place and the blue pigment is not going to be produced. For the one at the bottom, another metabolic or biochemical pathway, different substrate that we're starting with, it's going to result in the yellow pigmentation being produced. And similarly, there is an enzyme that is necessary for this reaction to take place. But in order for this enzyme to be produced, you need to have the correct genetic code, and that involves at least one capital Y. So what about the individuals that are green in color? Well, if these individuals have at least one capital B and one capital Y, there are instructions for both the blue pigmentation and the yellow pigmentation. These are then going to mix together. So again, these are individuals that have at least one capital B, at least one capital Y, and they are going to be the green in color. So finally, how do we end up with the white coloration? Well, in the case of the white, what that means is there's no capital B, there is no capital Y, there are no instructions to make this enzyme, there are no instructions to make this enzyme, and if you can't make either of these enzymes, well, you can't convert the substrates into the products, the blue and the yellow pigmentation, so that's what results in the white coloration and those parakeets. So again, this is polygenic because we have two different genes, again, just calling them the B gene and the Y gene, that are both contributing to the same thing, and that is the bird color. This one here, epistatic interactions. This is again where we do have more than one gene that's having an effect on the same trait, but this one's a little bit different because it's not necessarily a blending that we saw with the previous examples. In this case here, one of the genes or the products from one of the genes is actually going to interfere with, and most of the time what that means is it's going to mask the expression of the other gene. So it's going to in fact block the functioning of that second gene. And this quite often does result in non-Mendelian ratios, things that we wouldn't necessarily expect and predict from our Punnett square. So just as a reminder, with the two trait crosses, the ones that we talked about that Gregor Mendel would have done, for example, taking a look at the flower position, terminal or axial, and the plant height, either tall or short, these ones here are on different chromosomes. And not only of that, they don't affect each other. So the flower position has no influence whatsoever on the height of the plant and vice versa. These traits, these genes do not affect each other. But sometimes the genes do. So now for this one here, the flower color, what I'm saying is that we have three different genes. I'm just numbering them gene 1, 2, and 3. And I give you in each case two different alleles for each one of these. So we could write down several different genotypes who would have to have a combination of 6 different letters, 2 of the P's, 2 of the D's, and 2 of the C's. So let's just jot down some of these here. So if we take a look at this individual here, they have the instructions for the capital P, so they would be purple in color. Now this flower here has the instructions for dark, so it will be a dark purple color. And if we throw in a capital C here, that is going to allow for that coloration. And what we're going to see is dark purple for the flower color. But it's with the white that we can get a number of different possibilities. So we can get a white flower by, of course, having 2 lowercase p's. It doesn't really matter what the next 2 letters are for the D's because you can't have dark and light white in color. It is just going to be white in color. And it doesn't matter also what the ones are for the C. It's going to be the white that we're going to see for this particular flower color. But there are some other possibilities. What if there are instructions for the purple coloration, whether it's homozygous or heterozygous? What if there are instructions for the dark pigmentation? If you don't have a capital C, then that purple is not going to be expressed. In other words, that gene for the pigmentation, the purple pigmentation, it is going to be blocked if there is no capital C. So no color is recessive, but if there are 2 lowercase c's, it doesn't matter what the instructions are for the other two genes. This one is also going to end up being white in color. So this is an epistatic interaction because it is really this gene here, gene number 3, that's having an impact primarily on this one. So if there is no capital C, that information is blocked. The purple pigmentation will not be produced, regardless of whether those instructions are present or not. And we'll finish it off just by taking a look at this example here with the fruit color in squash. So in this case here, again, we have two different genes. And we have, first of all, that color in this case is recessive to the no color. Color is recessive to no color. So no color is really white. That's what we're talking about. So in terms of the letters, we'll use a capital W for the white. That one is dominant for no color. In order to get color, they would have to be homozygous for the recessive one. So that is one gene located on one chromosome at one specific location, but now there is a second gene as well. So this second gene is for the color, and we see that the yellow is dominant to green, so we'll use a capital Y for the yellow, and we'll use the lowercase y for the green. So if we're setting this up as a two-trait or a dihybrid cross, and in this case, if it is selfed, if we have the dihybrid, both of them are heterozygous, and you're crossing them, this is showing the different possible phenotypes that we're going to get. And notice here that it's not a 9 to 3 to 3 to 1 ratio. It's not the typical Mendelian ratio that we get here, but in fact, it is a 12 to 3 to 1 ratio. So I'm not going to draw the entire Punnett square for this one, but let's just take a look at a sampling of some of the different genotypes that we could get here. So why is it a 12 and not the 9 to 3 to 3 to 1? Well, there are different ways that you can end up with the white coloration. So it could either be that there is the capital W with the capital Y's, or also the capital W and the lowercase y's. So as long as there is the capital W that we are seeing here, that's going to result in the white coloration. It doesn't matter if it's 1 or 2. So from the Punnett square, if you were to draw that out and take a look at all of the different genotypes that fit into these two categories, well, that gives us our 12 of them that are going to end up being white in color. How do you get the yellow coloration? Well, yellow again is dominant, so all you need is one capital Y, but you can't have a capital W. They have to be lowercase w's to get any color other than white. So if we have two lowercase w's and at least one capital Y, that's where you get the yellow. So that's going to be the 3. And the final one that we get here, in order to get the green, it's only 1 out of the 16 squares in the Punnett square, because you have to have homozygous recessive for both of these. So you cannot have the instructions for the yellow coloration, and you also cannot have a capital W, which is for the white. So the only way that you're going to get the green here is if it is homozygous recessive for both, and that is only going to be one of the squares out of the 16 different possibilities.